This meeting is being recorded.
Okay, thanks. Nancy Leland is from the UNH Freshwater Biology Lab. She has been with us before. Some of you might know her. And she has been working with the commission with the island-wide cyanobacteria monitoring that we did. So she's going to go over what we did last summer and our plans for this summer, which will be really exciting. Okay. Thanks, Nancy.
Yep, thanks, Sherry. Yeah, so hello, everyone, and welcome to today's meeting. Sherry invited me to give you an overview of what we accomplished last year when we started the project and where we're headed with it. So we're pretty excited about our findings. So for today, title of my talk is The Cyanobacteria of Martha's Vineyard. We are looking at diverse habitats, populations, and toxicity profiles. And for some of you, if you don't know what I do for my work, I spend a lot of time up at University of New Hampshire at the Center for Freshwater Biology and Ecotoxicology. Basically what we do is working with cyanobacteria and toxins in different types of aquatic habitats. So today I'm going to be giving you an overview of cyanobacteria for those of you that might not be that familiar with it, and introduce you to the world of cyanobacteria, their population ecology and ecotoxicology of the populations. I'd like to provide you with some information on the cyanocasting model, which is what we used out on the vineyard last summer, and ecological indicators that are associated with that model, and then go into a little more detail on what we learned from last year. So some of the common cyanobacteria that we see in New England waters, we like to break them into different groups depending upon their general size, and you'll see why that's really important to us. But we basically call them two different groups. We have the picocyanobacteria, which tend to be very small. And what you see here are some of the size ranges of either the single cell forms, which is the Synechococcus. And then also we have colonies, colonial forms of these small cell organisms, ranging up to about 50 microns in size. So they're pretty tiny. Some of these colonies that are formed of these little, very small cells, you can actually see with a light microscope. So they're macroscopic. And then we have others, the bloom-forming cyanobacteria, which tend to be rather large and can get pretty big in size. And those tend to be greater than 50 microns in size. And you'll see why we use this sort of 50 micron measurement that we use to discriminate between these different populations. So in terms of their ecology, we tend to see a separation in terms of how these cyanobacteria function. So the smaller forms, the Synechococcus and the Aphanocapsa and Warnicinia, these tend to be planktonic in nature, and they lead to problems typically associated with transparency issues in water bodies, whereas our bloom-forming cyanobacteria, which have the gas vesicles, they're buoyant, and they tend to form the scums that we see accumulating along the shoreline. So both of these types of situations, problems with transparency and scums, can be referred to as bloom conditions. In terms of looking at some of the variables and different types of things that we think about with these different cyanobacteria, we have different morphologies with them. They can range from single cells up to large colonies and filaments. They have a distinct size distribution associated with them, ranging from 2 microns, which is not microscopic. You have to use other means to see them. So they're a, sort of a class of their own, up to thousands of microns in size. We definitely have some seasonal preferences in terms of when we would typically see these in the water bodies. And they're really, really good at exploiting different types of ecological niches. So as you're working with different types of environments that are subject to different light regimens, temperature profiles, nutrient concentrations, and salinity as well, you will tend to find different types of populations of cyanobacteria. And their growth habits are very different from each other as well. So these are the things that we think about when we're working with the cyanobacterial populations. And we also have some common cyanotoxins that they produce, and I'm going to go over those with you so you can be familiar with that. And that's the ecotoxicology part of what we do. So for the work we do here in the lab, we typically focus on 3 different cyanotoxins that are produced by these guys. The first one is the BMAA, the B-methylalanine, and the anatoxin A. And you'll see that I have these organized where the BMAA and the anatoxin are more aligned up with these picocyanobacteria because those are the toxins that we typically see being produced by the picocyanobacteria. And then as you move towards bloom-forming cyanobacteria, you can also have the anatoxin A and the microcystins as well. So one of the things that's really important to keep in mind when you're working with the cyanobacteria is that we do have genus and sometimes species preferences for production of these toxins, but multiple toxins are commonplace that are produced by them. So it's one of the sort of guideposts that we have to think about all the time when we're working with them. The cyanotoxins themselves are highly water soluble. They have very distinct modes of action in terms of how they can affect a receptor, whether that's a pet, shellfish, fish, humans, things like that. So we have acute and chronic exposure profiles associated with them. All of these cyanotoxins typically have individual degradation profiles to them, ranging from hours to weeks. Their bioavailability is very distinct, so we can have edible and inedible forms determining whether they are even getting into things like the food chain. And they have very different bioaccumulation profiles to them. So we can look and see whether we're having situations of what we call biodilution and also biomagnification on the other end of the spectrum of bioaccumulation. So all of these things we have to think about as we're working with these cyanobacteria. So needless to say, they keep us on our toes as we're doing all of our monitoring and assessment. But there are some additional research topics that need to be explored. We really don't know a lot about the human health and ecological risks associated with the picocyanobacteria. That's an area that's being quite heavily studied right now, and we are working on that as well. We still don't know the triggers of toxin production, so you can have a toxigenic cyanobacteria in your water body, but it's not producing the toxin. And we still, after all these many years, do not know specifically what those triggers are for toxin production. And one of the things that we keep in mind is, what's the purpose of the toxin production anyways, as far as the cyanobacteria is concerned? Obviously, they're not producing the toxins because of us, because these organisms have been around for billions of years, but there are several theories about it. One of them is called the chemical defense theory. There's also ideas that some of these may be micronutrient scavengers that help support the growth of these cyanobacteria. And then also that the cyanotoxins themselves, because they're highly rich in nitrogen, could potentially be a nutrient source for them. So the cyanopasting model that we use from this lab, it was developed here. We look at several primary factors to begin with to start to understand our populations. And the most important one is the community composition and size structure. which I already sort of went over with you, but we like to see what's there. And then we break it into the different size fractions to be able to describe how much is there. And that leads us into the conversation about the biomass of the cyanobacteria. So how much is that actually there in terms of its biomass concentration relative to everything else that's either within that population of cyanobacteria or within the water column with everything else. And then the third important factor, then we can talk about the cyanotoxin concentrations themselves. So as an overview, what I'm showing you here are the different genus that we typically find and have found out in Martha's Vineyard. Microcystis is very common, Dolichospermum, Aphanicapsa, and Synechococcus. The Microcystis and Dolichospermum tend to be the bloom-forming cyanobacteria, so they're quite large. Colonies tend to be floating and buoyant. The Aphanicapsa and Synechococcus are the picocyanobacteria, so they tend to be quite small. And as I said, we have, colonies, filaments, and single cells as well. We also have different methods that we use to identify them. Microscopy with a light microscope and pigment analysis using our fluorometers is really good for working with BFCs and even these colonial picos. But when you get down to the single cell picocyanobacteria like Synechococcus, you basically have to work with what's called epifluorescence or flow cytometry and qPCR. So now you're into a whole different realm of laboratory analysis to confirm what's going on with that part of the population. So one of the interesting things that we think about in the lab is that the ecological niches that these guys inhabit suggest their use as indicator organisms. And that's what we're building our program around. The BFCs in particular, they are very slow growing organisms and they have very defined specialized niches. And when you look at something like the picocyanobacteria, they are very fast growing. They are highly adaptive and they actually are so diversified in their niches. They actually are more of an exploitive type of an organism. So you can see response to a change in some type of a habitat and have the picos responding within a matter of hours to those changing conditions. So that's one of the reasons why we like working with picocyanobacteria, but they also present unique challenges. But it makes for very interesting study in science because things are happening so fast with this type of community. So the methods that I had mentioned previously, we use light microscope, and we're able to look at composition and dominance of the bloom-forming cyanobacteria to the genus level with the light microscope. With our fluorometry, we can do fractionation of samples and we can describe these unique populations. So we're basically able to break them into groups by doing the fractionation, so we can have discussions about our BFC samples, our less than 50 micron size samples. And within that, just that information from the light microscope and the fluorometry, we are able to have some ecological indicators that we work with, and we can address some of the issues that may be associated with them. And in particular, we can look at the growth rates of these populations, what's happening to them in terms of the response to things like nutrient inputs or changes in the light and temperature. We can also estimate toxin concentrations from that, and we can also look at trophic influences. And so that's what makes this program really great, is that we have very simple methods that we're using, and we're able to describe in really good detail what's happening with these different ecological indicators. By including picocyanobacteria, which is less than 5 microns in size, we're able to expand what we're looking at with our ecological indicators. And we also are able to more fully use the whole idea of accessory pigments. So one of the more common accessory pigments that people use is phycocyanin, and that is providing a measure of cyanobacterial biomass, as compared to something like chlorophyll that's going to be looking at all plant growth within the water column. Phycocyanin is cyanobacterial specific. The other accessory pigment that we're using is phycoerythrin. And the reason why we have chosen to use that is that that particular accessory pigment is pretty much specific for picocyanobacterial biomass. So we're able to discriminate what's going on within the sample and the different populations just using fluorometry. And one of the nice things about including the picocyanobacteria is we are able to have a more robust conversation about what's happening with the potential impact to the food webs, and also the process of aerosolization. So the cyanocasting model also relies upon the community composition as the most important variable that we use to initiate our analysis. And what we're doing with that is we're doing something that's called pigment fingerprinting. And what we're able to do is to describe the diversity of the populations based upon the pigment fingerprints. We're able to look at the biomass growth rates, toxin concentrations, and trophic influences just by using this fingerprinting approach. And we've used this in freshwater applications for the past, it's been 8 years now. It's very easy to use, it's low cost, it's reliable, repeatable, and also transferable. So that's one of the things we really love about this concept of the pigment fingerprinting. In the brackish systems of Martha's Vineyard, we are hoping that this fingerprinting could replace more expensive techniques for analysis of picocyanobacteria. So the fingerprints can take the place of what you might need to do for epifluorescence, the flow cytometry, and the qPCR, and still be able to have these great conversations about food webs and aerosolization. So our work that we did in 2021 down at Martha's Vineyard in conjunction with Sherry and company at the Martha's Vineyard Commission, we did apply the cyanocasting model at over 30 sites across the island. We were using light microscopy and fluorometry. And one of the major findings was that we did see community composition varied with salinity, enough so that we could basically put them into 3 separate groups. When we have systems that are less than 1 part per 1,000 for salinity, we have our BFCs, so our bloom forming cyanobacteria were present in all of the systems. Between 1 and 15 parts per 1,000, we were able to identify some halo tolerant BFCs at some of the sites. And once we got above 15 parts per 1,000, the picos totally dominated the cyanobacterial biomass. So basically the BFCs become absent in those sites that have greater than 15 parts per 1,000. And as I explained earlier, what this does is this creates an entire shift in terms of what you might expect to see for different types of toxin in your water samples. So the red stars that you see here, these are the sites that were greater than 15 parts per 1,000 that we monitored last summer. And the picos, as I said, they dominate the cyanobacterial biomass at these sites. So that was more than 50% of the sites that we studied last year. So obviously they play a very large role in our monitoring program and are important to our interpretations. Also for our ecological indicators, one of the things that we worked on in great deal is what was going on with our toxin concentrations. So we actually did work with the 3 different the cyanotoxins for analysis, the microcystins, the anatoxin A, and the BMAA. So let's talk about the toxin concentrations a little bit. What we know already is that we do see a correlation between what's going on with the amount of biomass as measured by the phycocyanin and different toxin concentrations. So we've been able to establish that and document that in freshwater systems for the past 6 years in New York, New Hampshire, and Massachusetts. We know that this differentiates based upon the community composition. So knowing what's there is very important. And it also determines what the toxin concentrations are. We've repeatedly verified this relationship for microcystins, and our preliminary studies for anatoxin A and BMAA suggest that we have similar dynamics going on. So the Martha's Vineyard project was the first study for brackish systems. So what I'm going to share with you is some of the preliminary results that we had from last year's survey work. So anatoxin A in our freshwater site, so that was 0.2 parts per thousand. We were able to work with not only the phycocyanin biomass, but the phycoerythrin as well for anatoxin A. And we were able to establish linear relationships between these 2 variables. So we basically were confirming this relationship between biomass and cyanotoxin concentration in our freshwater site. In our brackish site, we were not able to statistically say that we had a linear relationship, but we were able to see correlations between our pigment measurements, the phycocyanin and the phycoerythrin, and our anatoxin A concentrations. So the question becomes whether we need additional information about the picocyanobacterial populations, because we know that we're at these brackish sites, whether that's going to help with our analysis and get us from the place of just seeing correlations between these 2 variables and whether we're actually able to see some type of a linear relationship. So for our BMAA results for our toxin concentration, again, at the freshwater site, we were able to establish linear relationships between our cyanobacterial biomass using our phycocyanin and our phycoerythrin and our BMAA concentrations. And we were able to verify that with linear relationships again. So that was pretty exciting. At our brackish site, again, we were faced with the same question. We have correlations between the phycocyanin and the phycoerythrin and the BMAA. We would prefer just to know whether we actually have linear relationships there. So that's something else that we need to spend more time studying. So as far as picocyanobacteria and how they fit into our monitoring strategy to be a strong ecological indicator of toxin concentration in freshwater systems, our interpretation may be enhanced through the use of picocyanobacteria, but in brackish systems, from our perspective, our interpretations require the inclusion of picocyanobacteria. And the one thing that would be nice about including them, because it means extra work in our monitoring program, that we do allow ourselves to open the door to other types of assessments. And again, we're working into the world of food webs, bioaccumulation, transfer through the food webs, and also aerosols. And if any of you are interested in what we're doing with that this summer, I'd suggest you get in touch with Sherry, since she is the one who is heading up that program. So what's ahead for this summer, 2022, out on the Vineyard. Like I mentioned, we're going to be continuing doing the cyanocasting model and hopefully building off of what we learned in 2021 and potentially repeating some of those sites. We also have some special projects that we are going to be doing in 2022 that involve monitoring for cyanobacteria. We're going to be using our pigments again, phycocyanin and phycoerythrin. We're going to be looking at the 3 different toxins, the microcystins, the anatoxin A, and the BMAA. We are going to be using picocyanobacteria as a response variable to measure the effectiveness of stormwater control measures. So that's part of the Green and Resilient Infrastructure Project that's ongoing on the island. We have our sites are selected. We're going to be looking at Tajmu, the culvert site and the Sentinel site, and Lagoon Pond. And we're working with the UNH Stormwater Center through the commission, and then also this is funded through EPA. Another project that we're working on is an eDNA project. So we will be looking specifically at the picocyanobacterial pigment fingerprinting, its toxin production, and species identification. We do not know yet whether some of these toxin production or cyanotoxins that are produced are associated with particular species of picocyanobacteria, or whether we can make some generalizations about them to the genus level. So what we will be doing is investigating this Chilmark Pond, so Chilmark Up and Chilmark 2, Tisbury Great Pond, James Pond, Squibnocket, and then still at Tajmu and Lagoon Pond. And we are partnering again with the UNH Stormwater Center and USGS for this project. So we're pretty excited about that. It's going to be an interesting challenge, but I know that we're going to be learning a lot, and we're hoping that we get some really great results on that. So summary of findings. And basically what we're seeing are a diversity of cyanobacterial populations depending upon the salinity profiles that you have. So since you have such an extensive gradient out there on the Vineyard, you would expect to see many different types of populations all within a very small space. That we did see correlations between the cyanobacterial biomass and cyanotoxins even in the brackish waters. So that was pretty exciting for us to be able to confirm that, but we would like to investigate that a little more. It's obvious to us that picocyanobacteria play a very important role in the toxicity profiles. So that's something that we think deserves a lot more attention and research into, and that there's obviously opportunities to expand our array of ecological assessments that we can do using this method. So we're really excited about the possibilities. We're excited about what we've found so far, but we're really looking forward to what's going to be happening for this summer. So if you have any questions, I'd be happy to take them.
Looks like you did such a great job covering it all that there aren't any questions, which I'm amazed. Usually this group is full of questions. John has a hand up.
Maybe they just haven't downloaded all that information yet.
Yeah, they're still processing. John has a question. Go ahead, John.
Hi, good afternoon, everybody, and thank you, Nancy. That was extremely interesting. I am wondering, you noted what is the significance of the toxins to the bacteria? And I'm curious, I was curious about that before you got to it, you know, whither this, why, what is the adaptive value to these bacteria? And I guess, I mean, you suggested 3 possibilities. And I'm wondering if there's any research underway to further elucidate these. And I'm also wondering what your perspective is that if that research were done and you had a much better idea about the adaptive significance to these bacteria of toxin production, how do you see that would help you in going about your work and in dealing with
Right.
this?
Yeah, so it
Thanks.
would be really helpful because it can drive, well, of course, it would help us with our interpretations of when we see the values and they're changing and we're trying to make sense of, you know, what is it that's happening within the population. It can help us structure our collection methods so that we're actually doing a better job of doing the collections, so that if there's some questions in there, we know what we're supposed to be looking for rather than just assuming that we've already figured this all out. For example, microcystin, even though it's a secondary metabolite, we firmly, you know, feel that it does have a lot to do with growth, just because of what we're seeing in terms of the toxin concentrations during the times when we have high growth. And of course, that could tell us something about when do we have the greatest amount of exposure to someone who could potentially be coming in contact with these toxins, right? So knowing more about what the toxins are for the cyanobacteria helps us zero in on when are those times when we have the highest toxin production for whatever reason that is. And these are the times when maybe you don't want to be exposed to it through recreational activities. Perhaps there is a concern in terms of having these toxins ending up, you know, say potentially within filter feeders or something like that. So yeah, so that it's a real fundamental question that drives what we do, because if we can understand that better, we can understand how we're supposed to be monitoring, which then could translate into a better monitoring strategy to protect public and environmental health. It's a pretty simple equation, but it's not that easy to answer.
Another question, please. I heard first that in order to detect picocyanobacteria, you need really fancy technology like flow cytometry. And then I heard that doing the pigment fingerprinting, you could detect picocyanobacteria. So
Yep.
is the bottom line that we do have a method to monitor picocyanobacteria here using the technology we have available, or not?
Well, that's actually what we're working on this summer. So we have developed a new collection method based upon what we learned out at the Vineyard last summer, that we, and it's part of the fractionation that we do. And we submitted that collection methodology into EPA, and they have approved it as part of our QAP. So we will be implementing that, and Sherry has been trained on it. It's a little bit extra work, but we think there's a lot of value added within it, and we should be able to just use the handheld fluorometry to be able to get that information, and then it's a matter of interpretation of the fingerprints. And that's the real challenge for us this summer, is to be able to verify that. We have a lot of data from last summer, but we need to verify it. And that's why we're doing the eDNA project.
Thank you.
May I ask a follow-up?
Sure.
Thanks. So I'm coming at this as someone who used to do pure behavioral ecology myself, and I would always argue that it's necessary to understand what a critter is doing and why it is doing it if one's going to get involved in any aspect, in my case, of conservation and so on. And so in your case, given what you said, is the amount of support for that research commensurate with its significance to you practically, of having that understood?
We could always use more support. But in some ways, too much support can be, at the wrong time, can be not a good thing. So we like to walk before we run. So we felt like we did a lot of good walking last summer. And I feel like we made some pretty incredible progress. And that's why this summer we're really excited about where we're going, because we have some real burning questions around that, but we think that we will be finding some answers so that we can then move the whole program forward at a much greater pace than we might otherwise. So there's a lot of potential there, but we're making sure that we dot our I's and cross our T's on our methods and refine our questions so that we feel comfortable with whatever findings we come up with.
Nancy has been very good with getting me, keeping me under control and not trying to take on too much and just doing what we're doing well and not get overwhelmed. Because this could easily just take on a life of its own, but we all have other things going on also. So.
Yeah, I think it's really important to make sure that the foundation is as rock solid as it can be in terms of the science and the methods. And then the other challenge too is, this is being put in the hands of citizen monitors. Okay, so we are going to rely very heavily upon them to do the data collection, and Sherry is pretty much functioning as a satellite lab for us this summer. So she has to, do all of the sample processing and meet all of our QA/QC requirements, and, follow the new QAP and anything else we happen to throw at her. So, we have to be really careful how we proceed, but I think it's got a tremendous amount of potential, especially if we can have really good conversations about using these other environmental indicators and looking at, different exposure pathways, different types of risk assessments, and do it for relatively a low cost, but providing very robust information, and especially in terms of being able to, in the long term, save money because we're working with the toxins as well.
Okay. Does anyone else? John? John has another question.
I had another
That's
question
okay.
that's slightly different.
Great, thanks,
I
John.
noticed that I'm looking at a tiny laptop screen with your tiny map, but it looked as if you had some dots on the Vineyard map that, to my eye, were basically more or less to the east of James Pond. So I'm wondering, does that relate to your freshwater site or sites?
We did have sites. Yeah, there was several of them in the group of less than 1 part per thousand. So yeah, I think that's what Mink Meadows, Sherry.
Ice House and Cess Pond we looked at last summer. Land Bank collected the samples for us from Ice House, and so we looked at that. Those were our freshwater systems, and I don't think we found anything in any of those, or they were. Seth followed the regression from Lower Mill Pond from the Cape, correct, Nancy?
Yes.
Because Seth was one of the regression sites.
Yep.
Mm-hmm. So at this point, I think we're just going to continue the work in the coastal ponds. We're not going to branch out into the freshwater ponds. I'm trying to get a study together to look at the freshwater ponds, but that won't be. And that might be one thing that we look at for the cyanobacteria, but that's going to be a big project that we would definitely hire a consultant outside to look at all of our freshwater ponds, because there's so many of them.
Yeah, and I think that, like I said in my presentation, I mean, the cyanobacterial population itself is, you know, obviously you could spend a lifetime working just with picos. And there's, you know, they occupy so many, you know, such a large percentage of your sites are pico-driven systems. So to not, you know, delve into that in great detail and be, and to do a really good job at it is kind of like, wow, well, you've got to do your homework and you got to get the work done. So I'm okay, Sherry, just doing the higher salinity systems this year. We have a lot to learn about picos, but we're very, very excited about the prospects.
We'll definitely build on what we learned this year. We'll build on that and, you know, expand the program. And then we will be looking for more partners, John, and trying to branch out a little bit.
Understood.
Yeah, I'm sure we'll have all sorts of interesting questions coming up by the end of the summer. Presumably we'll even have questions before the end of the summer. I'm just assuming that we're going to be doing data analysis as we go along, since we have so many projects going on.
Does anyone have any more questions for Nancy? Because I see Adam has joined us, and he has a couple of things that he would like to announce that the commission's working on. Any more questions? John, you good?
For now.
We can always come back to Nancy if something comes up. You can just shoot me a message in the chat and I'll make sure that we come back to it. Okay, Adam, thanks for joining us. And Nancy, thank you so much for a great presentation. Every time I talk with you, I definitely learn more. It's great. So, and I look forward to the work that we do this summer.
Yeah, me too.
Adam?
Thank you. It's always tough to follow Nancy because she's always so organized and I'm always so not. But I think, Nancy, this time I do have you. There's a lot of exciting news over the last month, stemming from the different kinds of things that the commission is doing. We're really into the next phase of water quality. We do testing. Sherry is beginning her annual testing so that we do have a baseline. We are the certified baseline for the state. So we test exactly the same way that the MEP did, because the MEP is the state standard. But this year, first of all, we did get an ARPA grant, which we're finalizing with the county now, to provide $450,000 to island towns to basically design, not design, I would say more put in alternative wastewater systems, IA systems. We will work with the boards of health to do that. And we will basically put in as many as we can with the funding that we have. The boards of health and the health agents will develop the criteria for us distributing that. But again, we're into the nitrogen, getting rid of nitrogen diversion, we call it. And we are starting to get funds to begin to do that, moving from studying it to really making a move in places that don't have sewer. Second really exciting thing, and this is huge, is that the state, and we were involved in it, if you can see the footprints of what we're doing up island, has announced that they're going to revise both their Title V regulations and how money is given out of the revolving fund, which used to go to just sewer, that IA systems will be allowed, and other kinds of more rural efforts to do that, including Rick Carney oysters. So that's a huge step. This is the first time in 35 years that they've opened that up. If you, again, look at their language, using watershed development plans and other kinds of stuff, that's exactly what we're doing in the Up Island 208 plans. But it's huge that they have taken the step to standardize watershed planning and getting away from CWMPs, which were a more sewer-based sort of water quality management system, and more looking at watersheds and going through some of the steps that we're going through in the 208. So that's very exciting. I mean, for them to make that move, it's taken a couple of years. The Cape and Islands Water Quality Task Force was really involved in that too, the rural towns over there. So we're very excited. Third, as part of that, even more exciting, the governor, state of Massachusetts, will make $200 million available to wastewater quality projects through a state revolving fund for the Cape and Islands only, for places to implement watershed systems. Now, the Cape is going to get most of that. I don't want you to think that most of this stuff, a lot of the stuff's directed at the Cape. But the fact that, that there'll be millions of dollars for communities that have watershed management plans that are accepted by DEP means that they can be implemented. And again, they can be implemented in a lot of different things, including probably sewer, but definitely including alternative systems for rural places. So the resources are there all of a sudden to do a lot of different things to clean up the ponds. And I think that's the message, that this is sort of unprecedented. But you have to have these plans, and you have to have these plans approved. And the three Don Island towns are doing traditional CWMPs, which can be easily translated into watershed management plans. We're doing the 208s up island, which again, we're coming out with the existing conditions report, but those are really you've all seen that for 10 years. The question is, what are you going to do to clean up the ponds? And that's the second act of this, is looking at all the technologies. And then the third is developing a plan to address the TMDL and to begin to clean up these resources. So it's a very exciting time for us, and we look forward to. I said to somebody, if we don't take advantage of this potential, it's our fault, because the resources and the attention is there. Sherry, I'll take any kind of questions anybody wants to ask. If not. Jerry,
go ahead.
Adam, I heard yesterday that DEP is looking at opening up Title V to put standards in for nitrogen sensitive areas. Like, again, I think focused on the Cape and Islands. Do you have information on that?
I think you're right. It's basically those that they're the reason that two different letters were sent out is for the Cape and Islands. I think the DEP wants them to move quicker, and it's been 5 or 6 years that. So they've created sort of a new system of places that are really, really, nitrogen sensitive. And for us, I don't know that would, apply. It may, if we had areas that were that way, but I look at it more that we do watershed management plans. We come up with a series of solutions that we can be funded, that we can then go. Again, I look at it like cleaning up, go get our funds and actually clean these up. But I think that, Jerry, to answer your question, I think that's more directed at the Cape. All right, Sherry, thank you. Oh, John.
Adam, that's really amazing news. Great news. Thanks for sharing that. Has this been covered in the island papers?
I don't know. I'm not, I've not communicated. I know that all of the island towns got letters, as did I, from the DEP announcing it. I went to the DEP meeting where it was announced, and then went to the Cape and Islands. So it's certainly known throughout the island. I don't know if the newspapers have covered it.
I've seen it in an article in the Cape Cod Times, but I don't think I've seen it in the local papers. So that's a good point, John. We should send out the, kind of poke them. I'm sure they got the press release, but maybe they just didn't realize its importance or something. We'll try to give them a poke and see if they want to run something. Because it's huge, and all the towns will get IA systems. We have to set up a program, and we'll set up a monitoring program for them also, so we can. And the commission's going to be overseeing the database for monitoring to look at their effectiveness.
And one other thing I didn't mention is you're also going to have to come up with a plan to measure your effectiveness generally as to how much you're cleaning and other kinds of things as these things go in. Part of the Cape and Islands Trust, there's a research part of it that the Cape Cod Commission mainly does. They hired a consultant and are doing some stuff, and we did get included in that, so that it's called a community management entity or a structure of administration, and we're included in that as well. So how do we regulate these things? How does the towns jointly, collectively, individually look at these will also be covered. All right, well, thank you.
Thanks. Does anyone else have anything to add, or are we set to go today? Any more questions or comments or announcements? Okay, I'm not seeing anything. So does everyone think we should try to do a meeting in July just to keep in touch, or do we think we're going to be too busy? You can give me thumbs up, hands up, or thumbs down, or just pipe up on your. John, yeah, it's, I felt bad that we, COVID kind of threw us off. So I like to keep the momentum going. It's like if people, and with the recordings, it's really nice. If you can't make it, you can always check it, watch it later. So yeah, but we should definitely have some updates on the cyanobacteria. This summer, I have 4 interns, and they are all starting Monday, and we will be hitting the ground running. Next week, we'll be out on the ponds. Senjikataket is our first sample site, and we'll be doing water quality, and the volunteers of Friends of Senji will be collecting water samples for cyanobacteria monitoring. So we will be busy. We're going to do 13 ponds, and those will be sampled 3 to 4 times at least 3 times, mostly 4 on all the island ponds that we sample. So we'll be really busy, and we should keep you posted on what we're seeing. So, okay. If there's no questions, have an absolutely wonderful afternoon. And we'll see you the 3rd week in July, which is, that would be the 15th.
Thanks, Sherry.
Okay,
Thanks, Becky.
thank you, Sherry.
Okay, bye, everybody.
Thanks a lot, Sherry. Great
Sure.
news from Adam and
Good.
Nancy.
Yeah.
Take care of you all. And I look forward to the Gazette's coverage of you and your interns. Seriously,
Thanks.
it'll be good. Take care. Bye-bye.